Good evening, everyone, and thank you for joining us. Please welcome Michelle Kaiser, Columbia College, class of '87, Columbia Mailman School of Public Health, class of '92, Columbia University, Vagelos class of Physicians and Surgeons, class of '97.
Good evening and welcome to AlumniTALK, a virtual speaker series featuring prominent alumni who share their knowledge, insights, and wisdom with the college alumni community on important and critical subject matters. I'm Michelle Kaiser, Columbia College, class of 1987, Mailman School of Public Health, class of 1992, Vagelos College of Physicians and Surgeons, class of 1997, and a proud member of the Columbia College Alumni Association. On behalf of the college alumni community, I want to welcome you to an evening discussion on the efforts to fight COVID-19. Dr. George Yancopoulos, college class of 1980, Graduate School of Arts and Sciences, class of 1986, Columbia University Vagelos College of Physicians and Surgeons, class of 1987, in conversation with Columbia College Dean James Valentini. Before we get started, please note, if you'd like to participate in the Q&A during tonight's discussion, please enter your question, including your graduation year, into the Q&A box on the bottom of your screen. Your entry will only be visible to the facilitator. If your question is selected, we will ask your question on your behalf. Please submit questions at any time throughout the presentation. For optimal viewing, please enter full screen mode. Dr. George Yancopoulos is Regeneron Pharmaceuticals co-founder, president, and chief scientific officer. During the last 30 years, he has worked with his longtime partner, Dr. Leonard Schleifer, to build Regeneron into a leading science-driven biotech company. Regeneron has invented and developed in-house and in its own laboratories 7 FDA-approved medicines for serious diseases such as cancer, vision-threatening eye diseases, heart disease, atopic dermatitis, asthma, and rheumatoid arthritis. Dr. Yancopoulos is a unique biotech leader who has co-invented, along with key members of his team, Regeneron's foundational technologies for target and drug discovery, the TRAP, Velocigene, and Velocimmune platforms, as well as its breakthrough medical treatments, Eylea, Praluent, and Dupixent. The Regeneron team continues to lead biotech innovation, such as through the Regeneron Genetics Center, a world-leading effort that has already sequenced more than 700,000 people. Dean James Valentini has served as Dean of Columbia College and Vice President of Undergraduate Education since September of 2011. Affectionately called Dean Teeny by Columbia undergraduates, he has been a member of Columbia's faculty since 1990. Under his leadership, Columbia College has developed a multi-year strategic plan and launched a $750 million core to commencement campaign, the first campaign devoted exclusively to Columbia College students and faculty. The college has also initiated the Presidential Global Fellowship, offering a funded summer study abroad experience to rising sophomores, and expanded its commitment to entrepreneurship, joining the Columbia Startup Lab and sponsoring the Columbia Venture Competition's Undergraduate Challenge. A big welcome to our distinguished speakers.
Thank you, Michelle, for that very generous introduction. And we really appreciate it. And George, good to see you again.
Great to be here, Dean.
So the topic tonight obviously is COVID-19, but as Michelle's introduction of you indicates, you didn't just pop up on the scene a few months ago. So I'd like to start by asking something that's a little more general. Where and when did you get the inspiration for the work you do, which is part of your effort on COVID-19, and specifically, that is, where did you get the idea and when for Regeneron?
Well, I don't know if it was one particular moment in time, but for sure, it had its roots in my education, and there were a million different particulars that led to it. But I have to say it's the combination of all the mentors I had throughout my life. It was going to an incredible high school in New York City, public high school, the Bronx High School of Science, having incredible teachers there who inspired me to get involved in science projects. Once I got involved in actually doing hands-on science, I was hooked because you got to do things and you got to see cool results some of the time. And that just continued, as you heard, at Columbia, where I got involved from the early days in lab work. First, I had a couple different mentors there, from Tom Cass to Jonathan Greer, where I worked in their labs. And then it just kept continuing when I went to medical school and graduate school. And just doing lab work is in some ways addictive. There's a lot of failure. And I think it's sort of like golf, though I don't play golf. But I hear that, you hit that one shot perfectly every once in a while. And once you do that, you just want to keep swinging until you hit that shot again. It's the same thing in science, because most of it is failure. But every once in a while, you make an important realization, important discovery. You realize that you've discovered something fundamental about life that nobody else has realized before. And I think that there's something incredibly cool about that. And not only that, and the advantage and the reason I got involved in starting a biotech company was the realization not only could you make new discoveries about life and how life works, but you could take it to the next level. You could actually figure out how to take advantage of that and actually cure disease. And at some point that became the driving force in my life. I mean, I'm sure we've all encountered friends, family members, parents often, grandparents. And once you encounter disease and what it can do to somebody, if you believe that you have the power, if you believe you have the ability to actually make a difference in disease, and if you can actually change the course of disease, reverse it, cure it, that's an incredible potential ability. And I think it can drive a lot of people to devote their lives to trying to take advantage of that ability. And so that became, my reason for living is to understand life and fight back against disease.
Yeah, that's great. So you were obviously interested in science before you came to Columbia, but you have the distinction of very few people of having 5 degrees from Columbia: a bachelor's degree from Columbia College, an MA, an MPhil, and a PhD from the Graduate School of Arts and Sciences, and an MD from the Vagelos College of Physicians and Surgeons. What about that experience? How did that lead you to this focus on disease? Was this something that motivated you from early on, or was this something that developed over the course of time you were at Columbia?
I think it's all, I think all of us, we're all made up of all of the individual experiences that we had. And I certainly, like I said, it started for me at an early age, and then it definitely continued and it built on itself at Columbia from my earliest experiences. And they weren't always experiences of success. When I went to Columbia, I was very interested in structure of molecules, in chemistry and in physical chemistry, and at that time in what was known as X-ray crystallography. And I spent a couple of years working very hard in that field with very little success, essentially almost complete failure. And at one point, I'll never forget, I was in a lecture class that was actually being given by Charles Cantor. And they talked about, I'm so old, that this course was actually describing the cloning of the first human gene, which was a big milestone back in 1976 or 1977 or so. And it was incredible to me that we could literally isolate a human gene and identify it and actually see it was actually the hemoglobin gene, so you could actually define the genetic basis of, at that time, the disease known as sickle cell anemia and so forth. I became fascinated by that, but moreover, the names on the paper that they were describing in this class in biochemistry at Columbia, the names of the paper were Estradiatus, Maniatus, and Kaphatos. And it just really struck me. And this is how I guess life works sometimes. I had been struggling and failing in the field of structural chemistry. And then I see this great new milestone, cloning of the first human gene. The three authors of the paper were all Greek. And I said to myself, wow, maybe this is in my genes. Maybe I could actually become a cloner. And so I literally at that moment changed my interest in my field, and I became at the time what was known as a cloner. I ended up working with a lot of the giants at the time at Columbia, from Richard Axel to Fred Alt, who was my personal mentor. And it changed everything. The ability to clone genes, the ability to understand genes, the ability to manipulate genes was so exciting. It was a whole new science. It was a whole new opportunity. But that's sometimes how random chance and random events and experiences can change the direction that you're going in.
Yeah. So George, as Michelle's introduction of you indicates, Regeneron has had phenomenal success at producing therapeutics for disease, maybe more than anyone else. Was Regeneron an overnight success?
Yeah, great question. The answer is absolutely not. And in retrospect, I think that is actually a source of our strength. We burst on the scene about 5 to 10 years ago, and people referred to us as an overnight success. We came out of nowhere, it seemed to a lot of people. But what most people didn't know is that we had a history of 20 to 25 years before that of what a lot of people considered was complete failure. We did not, until about 20 years into our experience, have any success in the discovery and the development of new treatments. Over the last 10 years, as you said, we are arguably the most successful organization in the history of the world in terms of coming up with important new treatments. I think it was that failure. It was those 20 years where I don't really think we were failing. We were failing to the outside world, but the way I really look at it is we were learning. We were learning what wasn't working, and we were working how to adapt to that. And I keep telling people, there's nothing more important than that. Failure is not the end if you're trying to build on it and take it to the next level. And in fact, I am absolutely sure that the reason that I consider us to be the closest thing one could have in our industry to a bulletproof organization is simply because we made so many mistakes. We learned so much. We refused to give in. We refused to think that that failure was, a dead end. We just kept building and building and building. And eventually, after about 20, 25 years, we changed it. And people look at our success and they say, how is it possible that the rest of the industry has a 95% failure rate and these guys have a 95% success rate? Well, it's because we've been doing it for so long, and we learned because we had a 100% failure rate for about 20 years.
Yeah. So this brings us to the present moment. And so I want to turn to the pandemic. The pandemic has really brought into sharp focus many aspects of human existence. It made us aware of things that we had taken for granted. And it is, I think, showed us many, many challenges, but also some opportunities. How do you see this specifically in the domain of science and its connection to the human condition and human well-being?
Well, as you know, because we've talked about it before, many of us, not only me, it's nothing special about my viewpoint, I think we've seen these things coming. And by these things I'm talking about, we've seen an assortment of different truly existential threats against humanity. And they come in different forms. I strongly feel that, some of the forms will be in disease. Some of them have to do with what we've done to the environment. Some of them have to do with changes in the ways that we're interacting with each other. The threats of, to me, social media and supposedly the value of this interconnectedness, which is leading to so much polarization and divisiveness. I think that we are creating and we are contributing to an assortment of different existential threats against us. And I do believe that many of them are addressable with either science and technology or with the thought processes that go with successful science and technology. And so to me, I see this as a key moment in the history of humanity. Why? It's much bigger. I think it's much bigger than the pandemic. I am praying, I am hoping, and I'm doing my best that science and technology delivers here. Because if it can deliver here, I think it can reawaken us to those possibilities, that there are threats and we're not going to solve them by arguing amongst ourselves. We're not going to solve them by getting petty. The way we're going to solve them is taking advantage of the one thing that distinguishes us from all the other organisms on Earth, which is our ability to innovate and to think and to engineer ourselves out of the very problems that we've created. I mean, I very often have likened ours to bacteria growing in an Erlenmeyer flask. We've all done those sorts of experiments early on in our lives, or many of us have. When you grow bacteria in a tube or an Erlenmeyer flask, they just keep growing and growing and growing until they do two things. They poison their environment, which increases their chances of disease and death until they ultimately kill themselves. And frankly, we are no different than that on this planet Earth. Okay, we're just like those bacteria in a tube or an Erlenmeyer flask. And the one thing that distinguishes us from those bacteria is our ability to think. And I really hope that this is an opportunity for mankind to realize, this pandemic, that science and technology can provide a solution. And if we realize that, maybe we can start realizing that we should be investing a hell of a lot more in science and technology than we've investing to this point at so many levels, because it is really the only path to our salvation. Most people don't even realize it, but this pandemic has cost trillions, if not tens of trillions of dollars to the global economy. The thought that we as a nation have been investing about $30 billion a year in the NIH as an investment against disease, where one single disease here in a few months can cost tens of trillions of dollars, that's just bad math. That's bad investment. And like I said, I am praying that this is a wake-up call. And you know, you're a product of the same era, so you understand this. But I'm sure a lot of the younger alumni don't. There was different time in the world. I grew up in the '60s, and that was a time where I think it was a special time for inspiring young people to get involved in science and technology. And I think it certainly started for me at the top. We had incredible and inspiring leadership, perhaps one of the most inspiring presidents in the history of our nation, and certainly in terms of science and technology, JFK. And when he was assassinated, we spent the following year studying his speeches, and I recommend everybody, you know, go back and read them and look at the wisdom and so forth. When he proposed things like the moonshot, he actually predicted, he actually said, if you look in his speeches, it's not just about going to the moon. It's because we don't have the science and technology to get to the moon. To create the new science and technology that will get us to the moon will also simultaneously give us so many answers to so many other problems that we have as mankind. It will create so many new businesses, so many new jobs, and it will solve so many new problems and disease and so forth. And just that investment, it's not just about getting to the moon. The investment that we will make in the science and technologies will reap rewards that we can't even understand. And he talked about the fact that because those of us growing up at the time, we had different existential threats at the time, in some ways even more near term. We had the nuclear threat, the threat of Cold War. And I remember having to go, you know, we used to have the nuclear drills and we'd have to go to the bunker in the nearby buildings, you know, walking out of public school to prepare for the bombs falling. And he used to talk about how for the first time in history, mankind had the ability to both destroy the planet, but also save the planet. It was the power of science and technology, and that science and technology was not inherently good or evil. It was up to man to figure out whether they would use science and technology in a positive way that could save the world, or whether we would all lead to our own destruction. We are in that same position, except we don't have that sort of leadership that's inspiring us. Inspiring us, meaning the next generation of young people, to understand the power that they have. There's no bigger power than the God-given power to think, to engineer, to have ingenuity to get us out of our situations. And I think if we had that sort of leadership, young people would realize that it's not all doom and gloom. We can really make a difference. We can change ourselves. That's how I was inspired to try to cure disease. And I think we need that sort of inspiration. So I'm literally praying and hoping that we as a community deliver in this pandemic and show the world the path to our salvation, and change back the conversation, win back the narrative, and explain to people that science and technology can be providing the answers. It can provide the answers to, I think, all of our problems. We just have to make the investments, starting by recruiting and engaging the top young minds, that nothing is more important than giving back and figuring out how to solve all of our problems.
George, this isn't a natural lead to the question in this present moment about how, I'll call it, the biomedical research community is responding to this particular challenge, and how that's likely to lead to the kind of realization you just described by the public as a whole. Talk a little bit about the different kinds of therapies that are actually being addressed here, both generally and specifically at Regeneron, and maybe a little bit about the difference between therapeutic agents and actual vaccines.
Sure. And first, let me just say that I think a lot of people, like I do, are recognizing that this is a big moment in time for humanity and for our industry. Our industry has had, there's a lot of naysayers about the industry. Of course, there's been a lot of bad examples of bad behaviors, like in any industry. But that I believe that, we're all human. People are going to do bad things, but most people on average are going to be trying to do the right thing, and to paint any ethnic group, any race, or any collection of businesses as all inherently evil, I think is just, bias and prejudice and stereotyping. There's a lot of great people in the biopharma industry. And what I see now is they're all trying to come together. There's an incredible collaborative spirit, and people want to be heroes. I don't think there's anything wrong with that. I think that's what we should be. We should be challenging, the next generation to be the heroes that are going to save us from the problems that we've left them. And I think that right now, so many in our industry are trying to deliver, whether it's treatments or vaccines. And certainly we're in the midst of that. So just to give a little bit background for the current pandemic, what is a vaccine trying to do? What are our approaches trying to do? So I think that we all know or understand that when you get a viral infection, it doesn't matter whether it's the flu or COVID-19, the body's immune system comes into play, and it starts trying to generate what are known antiviral antibodies. That's the host defense. It's the body's defense against viruses. And these are proteins the body makes that will bind and kill the virus. And these are normally responsible, it doesn't matter, regardless of the virus, this is what the body uses to clear the initial viral infection. Once you've cleared the initial viral infection, which hopefully most people will do against most viruses, there are exceptions, AIDS being one of them, but it looks like it's true for COVID-19 that the body, on average, in general, makes these antiviral antibodies, clears the infection, and then you're left with these antibodies that the body continues to make that are a memory against this infection, that will protect against future infection by the same virus. And this protection can, in many cases, last your entire lifetime. That's why, historically, you get the chicken pox or the mumps or, measles as a child, you don't get it again in general. Well, what is a vaccine? A vaccine tricks the body into making a similar immune response as a real viral infection. It consists of not the real virus. You give either a dead virus, a weakened virus, or even a very small part of a virus called a protein or a piece of a protein. And the whole point is to try to elicit from the body, from the immune system, the same sort of long-lasting antiviral antibodies that will protect against the infection by you're making these antibodies that will bind and kill the virus. And vaccines range. Some of them can protect 100% of people from getting infected, and they can last a lifetime, like the measles vaccine. Very effective. Okay, other vaccines like the flu vaccine that you get every year is not that protective. It generally protects less than 50% of the vaccinated individuals, and it doesn't last a lifetime. So vaccines are generally an approach to try to mimic an infection to generate antiviral antibodies, but because they are manipulating your immune system, they're actually harder to make than regular drugs, because the vaccine is not the drug. The vaccine is trying to fool or trick your immune system to make the actual drug, which are the antibodies. So that's one of the reasons why, in general, it takes years, sometimes decades, to develop safe and effective vaccines. And I think we've all heard from Tony Fauci and others that maybe the earliest, if all goes well, despite record investments, there's more people working on this vaccine than have ever worked on any vaccine in history. And it still may take more than 1, 2, or more years to even be successful. And we may still fail. So what can you do in the meantime? Well, what some companies, what we've pioneered at Regeneron is these antiviral antibodies that I'm talking about, that you either get from a normal infection or that you can trick the body into making via a vaccine. Well, we've pioneered ways to make these sort of antibodies outside of the body. We've developed a whole assortment of technologies that makes the best of the antibodies that the immune system makes, but we don't make it in people. We make it outside of the body. We grow these antibodies up and we give them back to people as drugs. And these have been some of the most successful drugs now in history. Things like Eylea, which is the world's leading medicine to fight blindness due to diabetes or macular degeneration, or Dupixent, which is the world's leading medicine that can sometimes treat asthma and atopic dermatitis and other allergic diseases, or other ones that treat cancer and so forth. So we have this ability to make these antibodies, and we recently started applying them to infectious disease. And a major success that we had, which is one of the greatest successes in the history of using antibodies made outside of the body to treat infectious disease, was with Ebola. So a few years ago, during the last epidemic with Ebola, we had an effort that was spearheaded here at Regeneron by another Columbia alum, Christos Kyratsous, who was actually graduated from the same department that I graduated in terms of my PhD degree. And Christos led our effort with Ebola, and he created this collection of antibodies against the virus, which we called an antibody cocktail against Ebola. And it was dramatic. Normally, Ebola, we know, is much more universally fatal than almost any other viral disease, including COVID-19. And we were dramatically able to save lives. Much higher levels than, for example, these non-targeted drugs like remdesivir, which have already been tried, these repurposed drugs that have been tried for COVID-19. So anyway, since we've pioneered these antibody approaches, we've pioneered them for other classes of diseases outside of infectious disease, such as cancer or eye diseases or asthma. We started using them for infectious diseases, most notably Ebola. Incredible benefits seen there. So when the coronavirus epidemic hit, we put our resources on that. And we very rapidly created a similar antibody cocktail to what we had created for Ebola, but one specific for this virus. We put it into all sorts of preclinical testing, including in non-human primates. The results are very, very promising. And based on that, in record time, we put it into human trials for COVID-19 earlier this month. Less than 5 months from beginning the project to going into human trials, which is some sort of record in the industry. And it's in multiple trials, including to prevent infection in people who have not yet been infected, just like a vaccine could do. But unlike a vaccine, we can also give it to people who are already infected. That's what we showed with Ebola. We could take people who had about a 10 to 15% chance of living and increase that to 30 to 40%. So we're really hopeful that if we have similar results here, it could really make an impact both to help prevent COVID-19 in people who have been infected, but also treat people who are already infected, either in the early stages or the later stages. So I think there's real reason for hope based on where we are, that we're already in clinical trials, and that the approach had worked so well for a much more universally lethal disease that is Ebola.
So now I'm going to ask you to turn that expression of hope into something specific in terms of a prediction. So everyone wants to know, when they're going to be safe again. When can I take off a mask? When can I resume a normal life? When people must ask you this all the time, what's your projection, particularly given that your therapeutic agent is already in clinical trial? What's the timeline of your hope?
Yeah, I really, of course, like everybody else, I mean, we can hope for magic, that somehow it's all going to go away. And there is some science to suggest warmer weather, can handicap the spread of some viruses. But right now, if you look at the numbers, it's not looking that way. I mean, right now, as we've all heard, in some of the southern states, which had been spared up until now, they're having record-breaking levels of infections in the outbreak now. And it doesn't really make sense to me that we can open up until one of two things happens. Until this virus spreads so widely that there's what they call sufficient herd immunity. So many people will have had it that now we don't have to worry about it anymore. The problem is, if we let that happen, there may be an enormous amount of misery and death. So otherwise, to me, it seems like the only way that we can safely feel that we can open up and we can resume our lifestyle is if we either had a very effective vaccine or a very effective treatment. And a vaccine is a little bit of a while away still. So our treatment has a chance, I think, of making a difference because, as I said, we could find every person who is sick and treat them and make them uninfectious. And we can also potentially find and prevent the infection in all of their contacts in an approach that's known or been referred to as ring vaccination. So if you had an effective treatment that kept people from progressing and having to go on ventilators and getting really sick, while also stopping the infection in the ring of people around them, I think that we could literally put a stop to this pandemic. And it depends on having a really great treatment. So there's a hope. And things can always go wrong because biology, as I was saying before, is so complicated, and it always, you think you understand it, and there's always something that maybe you've missed. So things can still go wrong. But if all goes incredibly right, if the trials really proceed, if we've designed them right, if we start getting answers sooner rather than later, it's quite possible that within a month or two, we will know if this is effective and how effective it is at stopping people who are already infected from progressing to having to go on ventilators and so forth, as well as how effective it is at preventing somebody who's being exposed from even developing the infection. If our treatment within a month or two we know is highly effective in both those settings, that would be an opportunity to start giving it more widely. The FDA would have to review the data and agree it's worth giving it more widely. And then we would have a weapon. And we have such a weapon that can treat somebody who's infected and keep them from getting really sick, and also stop spreading locally the infection around that case. I think that can really change the course of this pandemic. People, I think the amount of fear and concern can go down. And I think that, we would have reason to feel as if we could really go back somewhat to a more normal lifestyle. Though I have a feeling that things are going to change, permanently in terms of some of the ways that we behave and act anyway.
Yes, I think we all believe that, but I think it'll be a great summer for all of us if in 2 months you can tell us that this is done well in clinical trials and it has that real promise. That would be fantastic. So, I mean, each of us has a different kind of experience of a realization of the problem of the pandemic. Mine, in some ways, is connected to the fact that on On the night of March 3rd, I had to cancel the John Jay Awards dinner, which was to be the following day, and call all the awardees up and say, I'm sorry, we're not going to be able to offer you an award because we think it's unsafe to have this John Jay dinner the next evening. That was a time of great uncertainty. Lots of people thought, well, you know, we shouldn't be doing this. And what was the evidence we should be doing it? A week later at Columbia, we converted our classes to online. 2 weeks later, we asked all the students in Columbia College and undergraduates in engineering to evacuate campus and go home. That's how that was my personal connection to the realization of the seriousness of this. What's yours? What were you thinking at that time? You have a much different perspective. Everyone has a unique perspective, but yours is a particularly significant one.
Well, I have to say that at first, it was just like so many other things that I work on. It was so theoretical. We were following, particularly, like I said, the person who leads all our efforts in this space, Christos Kyriatsous. And, you know, he was telling us about what was going on. And we started having a variety of interactions with scientists, both in Asia and then soon after that in Italy, where there was a huge outbreak. And I was literally on the phone talking to these people at the front lines, physician scientists in these places where the epidemic was breaking out. I could not believe the stories that they were telling me. And so we were preparing theoretically for this scourge to hit America and to hit New York. But part of me, it still was just, it was just intellectual. It was just theoretical. I just could not believe that what I'll never forget some conversation I was having with some physician scientists in Italy, where they were explaining to me that they literally were dealing with the most horrific situation that a doctor ever finds himself in, where they had so many patients, they had to choose who was going to go on a ventilator. And that meant that a lot of others were going to die. And I was hearing their words and I was hearing their desperation. And I just could not imagine that, and we were preparing for this, but I just could not really imagine that could possibly happen in this day and age in my city, in New York. And then it hit. And then all of a sudden, I started not only hearing the numbers, but I started getting the phone calls, you know, because I'm, you know, in my family and in my community, you know, I'm one of the few physicians and scientists. And all of a sudden, so many phone calls from so many people, you know, wives or friends of mine who are now on ventilators in New York City. And I like I said, that was an incredible, like you said, just the way you described, you know, one day you're thinking about whether you should not have this dinner. And then all of a sudden, you know, a few weeks later, you're switching to online classes. You know, I'm thinking about this theoretical problem and how we're going to solve it, and that we're moving along this potential therapeutic and all this. And then the next day, it's hitting my friends and relatives overseas. I mean, it traveled from Asia to Italy to my hometown. And I think that this is, you know, this is a great equalizer. And I think it just shows how we're all in it together. We are one mankind, one humanity. This is why I think it's such a great example for us. And it should unite us, which is why I'm a little disturbed with how there's other evidence that at the same time we're increasingly getting divided over so many different issues. When we realize that we're one humanity, that a single virus can travel within weeks to months from China through Europe and into America and cause such devastation, that's the enemy, guys. I mean, that's who we should all be getting together and fighting. The existential threats that should be pulling us together and making us come up with united effort against all of them, whether it's, you know, climate change in the environment or whether it's disease and the disease burden. And I'm, once again, I am really praying and hoping that somehow as a community of people, we start recognizing who the real enemies are. But to me, it was such a phenomenally depressing example of how vulnerable we are as a species and how we really need to come together to really fight for our salvation, because nobody else is doing it for us, and it's up to us. And like I said, I hope that we prove that science and technology can provide the answers and create a new beginning where we come together and we recognize who the real enemies are, and we unite as a species against the things that are threatening the actual survival of our species.
Yeah. So I had hoped to conclude my questions by asking you what you thought the lesson of the pandemic was, not how we responded to it, which people talk about it a lot, but it seems to me you just described something that I myself have thought, that it shows everyone is your neighbor, no matter how far away they may live from you, that everyone lives in your neighborhood, and that you're connected to everyone else in the world. And transportation, trade, and other things have reduced those physical distances, even of things like this, so that a disease that starts in one place in the world can be all over the world in no time at all. And we're all linked together. This virus has linked us all together in a way. Yeah. So
Absolutely, exactly. And like I said, I hope we learn some lessons here. I hope we learn that we are all in this together. We're in this game of, live or die all together as a species. And this is just an example of it. And the same is true whether it's climate change, or whether it's the next pandemic, or whether it's the diseases that we're not thinking enough about that are also achieving, unimaginable statuses and rates, not only developed and undeveloped countries. I mean, the rates of obesity and type 2 diabetes, they're going to dwarf the impact of this pandemic in just a few years. And we are not prepared for that. We don't have treatments. And essentially, whether it's more viral pandemics, which are going to come with increasing frequency just because of what you said, because of globalization, population density, but also the damage that we've done to the environment and to the biodiversity. So pandemics are going to come with increasing frequency, but just the regular old diseases that we haven't even addressed or cured are coming. And so I pray that we learn that lesson, that the real enemy is not each other, that we shouldn't be fighting each other, that we should be fighting together against the things that threaten our real existence as a species. I mean, the thing that I look, to me, frankly, and this, some people, may be offended by this, but to me, there is nothing more important than the immortality of our species. If there's no humans around in 100 or in 1,000 or in 1,000,000 years, all of our lives would have been in vain. The only thing that gives value to our lives, as far as I'm concerned, is that we continue to exist as a species. That is our route towards immortality. I hope that that's the lesson that we learn, that the only way we can be immortal as a species is by devoting the best and the brightest amongst us to addressing in a unified fashion all of these existential threats, most of which are of our making.
thank you, George. And there's a smaller scale thing in which we're all in this together, and that's tonight's event. And there are hundreds of people who are participating. So at this point, I'd like to relinquish my time to ask you questions and actually ask Nell, our producer, to come on and communicate to you questions from our audience. Nell.
Thank you, Dean Valentini. Our first question comes from Michael Brown, Columbia class of '69. Candidate vaccines will be tested first on young and healthy volunteers. Since COVID-19 disproportionately affects the older population, especially those with other health conditions, how will the risks and benefits to this population be assessed? And does this mean that a vaccine would be available to older patients only later? And if so, how much later?
Okay. George.
Okay, that is a great question. And that is true about vaccines, that in general, you have to go through these stages, and you have to start with the healthy and younger individuals. And those people's immune systems tend to be more robust. So vaccines tend to work better in those types of people than obviously the people who are most at risk from this virus, whom are the elderly and the immunocompromised. That is another advantage. So we all need a vaccine. And like I said, I am rooting for everybody in our ecosystem. And we ourselves are doing some things with vaccines that are further off. Other people are doing a lot of things with vaccines, some sooner, some later. But that's why there's also value to having so many different approaches, including our antiviral antibody effort. That approach actually does not have to be started in young and healthy individuals and volunteers. We're actually our initial trials, we're in people who are already sick, both people who were in the early stages of infection, but also people who are in the severe stages of the infection, already hospitalized. So that's the difference, because the primary point about a vaccine is safety first, and that's how you establish it and study it. But with our antiviral antibody drugs, you can literally go into the sickest and the most needy people first. And there's the potential that these antiviral antibodies will work even in people with no immune systems who are immunocompromised. So there's a lot of reason, a lot of hope for all the approaches that we're taking. And that's one of the reasons why these antibody cocktails that we're developing can be nearer term and can have a lot of use and utility for the most at-risk individuals, and long before we can do it with a vaccine.
Yeah, thank you, George. And I just want to point out Nell has questions already that have been submitted, but those of you in the audience, you can submit a question. There's a Q&A box at the bottom of your screen, which is the path by which you can submit a question. Nell, on to our second question.
Thank you. Our next question comes from Michael Anagnos from the Columbia class of 2018. Dr. Yancopoulos, do you have a view of whether COVID-19 cases in the US have become less severe since March and April? And if so, is this evidence of viral mutation or other factors?
Well, I have to say that I know Michael well. He's the son of a classmate of mine, George and Agnes's. So hey there, Michael. And that's a great question. I think that that on average, cases may be less severe or affecting people who are less vulnerable, in part just because of the natural way the epidemic has evolved in America. I think early on, we made, and I'm not blaming anybody, because I think that it's very hard, so I definitely do not think any of us should be in the blame game. It's very hard to know how to do the right thing and what the right thing is to do, especially when you're encountering something you've never encountered before. So this is not meant at all to be judgmental in terms of any of the decisions that were made. But early on, we made mistakes because we didn't really have this experience before. We didn't know what was going on. And in those situations, as I already explained, just like Regeneron did in its first 20 years, it's going to be a lot of failures. So early on, we did not do things to, for example, protect the elderly and those in nursing homes. Many of the early deaths and events were occurring in, for example, those who were hospitalized and in nursing homes, a very high percentage of the deaths. Now, I think because of the way spread is happening, it's tending to happen more among younger patients and people who are less at risk. So there is the appearance, that it may be less lethal now. I think that's just an artifact of how spread is happening now. I don't think that there's any evidence to suggest that the virus has substantially changed to this point, that it's become more or less infectious or that it's become more or less lethal. However, we know that once we invoke treatments, that the treatments will select for mutations and escape variants. That's what happened with AIDS. The early treatments for AIDS were very effective as individual agents, but very rapidly resulted in resistance. That's when you apply drugs, you're literally applying what's known as evolutionary pressure or selection on the virus, and you're going to select out the mutants that are resistant to your drugs. That's one of the reasons why we have to be very careful about the kinds of drugs and approaches that we test and how broadly we test them, because we don't want to be making huge, devastating population mistakes and selecting widely for escape variants. It's one of the reasons, I don't want to get into the details of it, that's why we're moving forward with what we call an antibody cocktail, because that tends to, and we've actually shown this in preclinical studies, it protects against viral escape and mutations. If we test what are known as single drugs or single antibody treatments those could have devastating effects in terms of seeding the landscape with escape mutants. So bottom line is, I do not think that right now there's much evidence that different versions of the virus are taking over. I do think that's going to be a much bigger concern once we start putting out effective treatments out there, because the nature of treatments and viruses is to select out for escape variants.
Nell?
Our next question comes from Daniel Shainholz from the Vagelos College of Physician and Surgeons class of '87. Can you hypothesize on why some individuals are more susceptible to COVID-19 and others are asymptomatic?
You know, that's a phenomenal question. We and others have been trying to understand it. This is also true for other viral infections, though obviously with so much focus on this disease and the devastation that it has caused, it has tremendously focused us on this question. There's been all sorts of theories, including ones that we've tested, including in the clinic, that have not necessarily turned out to be right so far. There was a theory that some people mounted this uncontrolled cytokine response. It was called cytokine storm, and that those people, because for whatever reason, whether it was genetic or acquired, they over-responded to the virus with cytokine storm, and particularly evidence initially coming out of both China and Italy was that it was driven by a particular cytokine known as interleukin-6. Some of you may know we were obligated, since we had one of the 2 IL-6 inhibitor drugs approved in the United States, we were obligated to test it. We did very large studies, invested a lot of time and resources, only to find out that blocking the cytokine storm, we very effectively blocked the cytokine storm, but we didn't seemingly help the patients out who were suffering from most of the stages of the disease. We still have one ongoing study there in the most critical of patients, but the results there are unlikely to be, black and white. So theories have already come and gone. Cytokine storm has been one of them, which right now is falling out of favor. We don't know, is the bottom line, and this shows how hard biology is. A lot of viruses behave this way, in that some people have much worse outcomes than others, and for most of them, we still haven't figured it out. So hopefully somebody figures this out real soon, if not else, somebody else, but it could be years, if ever, that we ever understand this.
Our next question.
Our next question is from Margaret Robinson, Columbia class of 1990. You mentioned using your drug to treat those exposed as well as infected. It seems that would require well-executed contact tracing. What are your thoughts given recent reports that contact tracing in New York, for example, is challenged?
Yeah, I think that maybe you're aware, for example, of the spectacular results they've had in some places like Iceland, identifying the initial case studies and all the contacts there and controlling the disease thereby. I think that obviously Iceland is a unique situation with a much smaller population and a much more defined way of following and accessing all individuals. I can imagine that logistically, New York City is a nightmare for trying to do this. And I think that, and I know that there is enormous efforts underway to try to take this on and to do it better. But as you said, especially the larger the population, the harder it is to define the population. The fact that not everybody is in a database that you can immediately access and follow. I mean, these are things, when we talk about lessons, I would think that would be an enormous one that we could learn, but it'd be an enormous challenge for the United States to do, to get a single integrated electronic health network database for every person in America. Somehow, I think it's going to be a while before we figure out how to get that done. But yes, it's going to be very hard. So for our studies, we are depending on that. And to a large degree, we're doing a lot of that ourselves. It's part of the process. As you said, executing these large clinical trials is a challenge. It takes an enormous investment in time, effort, and resources by people who are just as skilled and just as innovative as the people at the discovery end in our science. They are right now doing their best to do exactly what you said, which is figure out how to get the initial cases and to actually track and study and capture the contacts so that they can actually treat the contacts and in a well-controlled manner where they have placebo groups and so forth. So it's a big challenge. It requires a lot of expertise, a lot of resources, and a lot of ingenuity. And I'm hoping, and I think that our guys are up to the challenge.
Okay. More questions from the audience now?
Our next question comes from Bonnie Berger, Columbia class of 1999. Would you feel comfortable traveling this summer or this fall?
You know, these are really tough questions and everybody has to, figure out how to answer them individually. But like we said at the top, when Dean Valentini was asking me, I think that until we know that there is a treatment that you could give to somebody who is infected that dramatically decreases their odds of having to progress to a ventilator and then maybe dying, especially for older people who are at risk. I fall in that bad demographic where the odds are against you if you get sick. So I am very concerned, and I think a lot of people, depending on their demographic, should be very concerned. So until we have an effective treatment, I don't know how other people would answer that question, but I would choose to be cautious. I am praying and hoping that our treatment works. And if by the end of the summer, we know that you can take somebody who's infected, treat them with our antibody cocktail, and dramatically decrease the chances that they're going to have to progress and go on a ventilator and maybe worse. Up until that point, I think it pays to be very, very cautious. Now, whether we could have handled the epidemic differently, whether we could have different rules for different demographics, what a university should do for students as opposed to for professors. I haven't looked at the age-dependent numbers in the last few weeks, but up until about a month ago, less than 100 people under the age of 25 had died. There's over 100 million people under the age of 25 in America. That meant that if you looked at the numbers, you had much greater odds of dying in a car accident, even though people were driving a lot less because of the pandemic, or death due to so many other reasons than to COVID-19 if you were under the age of 25. If you were over the age of 65, your odds were 1 in 1,000 of dying. So it was less than 1 in a million to greater than 1 in 1,000. We treated everybody the same when we locked down society, when we shut everything down. Whether there was another reason, if we had had the time to really plan it, whether we really should have closed down universities, whether we really should have kept, people under the age of 25 under such restrictions, or if we could have instead figured out a way to allow them to maybe develop some herd immunity without communicating the possible disease to the people who are at high risk. I don't know if that was possible, but we treated everybody the same. But the facts is that everybody is not the same here. People under the age of 25 are at exceedingly low risk. People over the age of 25 are exceedingly high risk. And for those of us in the bad demographics, until there's a treatment, I think there's great danger to trying to just resume, travel and normal life.
Yeah. And George, you mentioned universities, and this is the this is the challenge where we at Columbia and other institutions are working with now. What should our university be like in the fall? We have both lots of young people, but we also have lots of not so young people, and they're joined together in a classroom and in many other domains. And the concern about all of them is and the complicated variation in risk among them is a difficult, makes decisions difficult.
Well, the irony of it is that I think by sending so many students home, we actually may have even exacerbated the situation. I know in my own household, I have 4 children, you know, all of them, well, 3 out of the 4 were in school, one of them at Columbia Law School, another one at Princeton, another one at Penn. And when they came home, one of them brought with them a friend who a few days into the experience turns out was infected.
Yeah.
And I think that happened all over America, that by sending young health, and she got over the infection within a couple of days with no untoward effects. But I think that it was one of probably millions of examples of young, healthy students who really had very low risk of anything bad happening to them, going home and spreading to parents or even worse, elderly grandparents, the risk. So what I'm saying, it was impossible to really plan it out. And I'm still not sure that I have a plan of how we could go about it. But I'm not sure that what we did was the best way of totally shutting down society. Not that I have the answers.
Yeah. But George will have an answer to the next question, Nell.
Excellent. The next question comes from Rachel Karp, Columbia class of 2010. Do you think that COVID treatments and the coming vaccine are/will be available to all and free at the point of delivery?
Yeah, it's another great question. And I do think that we would all love to live in a perfect world where everything was free and available to everybody. It turns out that there is no medical system in the world, okay, that can support the amount of disease we have without COVID-19, let alone in the face of COVID-19, and make it free and available to everybody. So in the case of our treatment, let us just say that vaccines are still years away, and our treatment is proven very safe and effective. That would sound great. But it turns out that we are one of the largest biomanufacturers in the world, probably one of the top 5 in the world. We could only make at most, somewhere on the order of 1 million doses a month. This is by devoting essentially all of our capacity to this, which would mean that we would stop making drugs that cure blindness, that cure cancer, that cure asthma, and so forth. What people don't realize is, in an ideal world, we want everything for free. We want everything available to everybody, okay? The problem is that, you know, just making a biological is so complex and it takes so much effort that it's just impossible. Now, certain kinds of vaccines, depending on which vaccine, are easier to make, and maybe we'll be able to make billions of those with time, not necessarily in the immediate future. So if we really want medicines to be much more broadly available at much reduced cost to many more people, as a society, we have to decide what it is that we want to invest in. Okay, these things are not going to come out of thin air. They're not going to appear by magic. They're going to require much more massive investment by society. And I don't know if society is ready, willing, and able to do that. Like I said, just for promoting the basic science that underlies a lot of the medical breakthroughs that we have to support the NIH. Okay, our society right now, our governments, okay, and the taxpayers are only providing about $30 billion a year to support the NIH and basic research that's done at the NIH. Okay, where if we look at the impact, there's been trillions to tens of trillions of dollars of impact of this pandemic on global economies. And so I'm sorry, you can't get things for free unless you're willing as a society to invest and pay a lot more. And we have to come to those, come up and deal with those hard questions. People often look at the biopharma industry as bad, evil players because they don't provide things for free. No, I'm sorry. Society has to decide to actually reward and create incentives even bigger for companies to invest even more, but it also has to decide to contribute in the basic science space, in terms of the NIH and so forth. I think it's a joke. We should be upping our investment in the basic sciences at the NIH tenfold or more. And we should be increasing the incentives for biopharma to be delivering even more. We have to come up with mechanisms to create a system where we actually have cures to most diseases. We don't have cures to most diseases. So forget this notion. This notion, I mean, it's a travesty. The notion about universal healthcare, healthcare for all, make everything free. You got to figure out how to incentivize society to be producing these cures and making them more broadly available. You can't get something for nothing. You got to increase the investment. It comes at the investment we make at the NIH, which got to go up, in my mind, at least tenfold. Okay, not 5% or 10% is what people are talking about. And we got to figure out how to incentivize businesses to invest more and to build more manufacturing space and so forth. And on top of that, I think we got to do a much better job of incentivizing and engaging the best and the brightest minds to get into these fields. What people don't understand is we don't have the cures for most things, okay? And they're not easy to come up with. And unless we can somehow explain to young people that there's nothing more important that they should be going into, figuring out how to cure, forget this pandemic, okay? There's a pandemic of obesity coming along. 25% of the US population, because of obesity, is within the next 20 years probably going to become type 2 diabetics. That means they're going to be losing limbs and kidneys and eyes and so forth. We're not prepared for that sort of devastation, and it can't come for free. It's going to have to come from much more investment from society, and people have to come to terms with that. And that means that well-meaning politicians have to understand that healthcare for all is a joke unless we understand how to come up with cures and how to create a society that will pay for those cures.
Nell?
Next question comes from Fei Lang, PS class of '68. Do you have thoughts on why children seem to be less susceptible to COVID-19?
Yeah, well, it's related to the question before about why some adults get very sick and some adults, are asymptomatic. These are great mysteries. It's not only true for this disease, but for an assortment of other diseases. I just mentioned type 2 diabetes. Some people have type 2 diabetes for decades and they don't lose their kidneys. They don't lose their limbs and so forth. And some of it is related to how well it's controlled, but some of it is related to other genetic and environmental factors that we still don't understand. This is why we need basic science. This is why we need much more investment in the NIH, because these are fundamental questions of disease that have to be answered if we really are going to come up with cures, not only for this pandemic, but for future pandemics. There's got to be fundamental investments in the science and technology to give these answers. So now we got this pandemic. It's focusing us all on these problems. Where do these answers come from? They come from people who spend enormous amounts of times usually failing at trying to answer these questions, which is why society has to be willing to invest a lot more in trying to get these answers so that we're not left here in the position that we're in here now, where we're all struggling right now, doing the best that we can to come up with these answers to questions that usually take decades to answer.
Okay, very good. Nell?
All right, and we want to thank everyone so much for their questions this evening. This will be our last question from Nonye Odukwe. Dr. Yancopoulos, how do you think the nation is or should be preparing for the potential second wave of COVID-19 in the upcoming flu season?
Yeah, these are all great questions. I think the best way it could prepare for it is to come up with safe and effective treatments. And I'm hoping that, for example, our treatment, like we were just describing earlier in the session, if we could essentially not give it to everybody, but give it to the people who are infected and also to their local contacts, there may be a way of turning millions of doses into something that can really help hundreds of millions of people. In lieu of that, of course, we're all waiting for things like vaccines that might be able to be manufactured and delivered even more broadly. Unless we have those safe and effective preventative or actual post-infectious treatments, then all we can do is keep doing, what we've been doing to date, which is practicing, social distancing, wearing masks, the hand washing, everything that we're taught, limiting, contacts in crowds. And also, of course, trying to do whatever we can to limit the inundation of healthcare resources. Like if there are outbreaks in particular areas, because people are not practicing these things, then the healthcare systems there will be completely flooded out, as we've seen happened in places like New York City, happened in Italy. So until we have treatments and or preventative approaches like vaccines, all we can do is continue to follow the sorts of advice that we've had from leaders like Tony Fauci and the CDC and so forth.
Thank you, George. That's a good closing advice for all of us. Thank you. So I want to thank the audience for the questions, and especially George for taking the time to participate in this discussion and make yourself available to so many of our alumni. In closing, I will like to introduce Alex Rosen, Columbia College class of 2011, who will deliver the closing remarks for this evening's event. Over to you, Alex.
Thanks, Dean Valentini. Hi, everyone. My name is Alex Rosen, Columbia College class of 2011 and President Emeritus of the Columbia College Young Alumni. I would like to take this opportunity to thank Dr. Yancopoulos and Dean Valentini for an evening of learning about the complexities and the progress being made in the efforts to fight COVID-19. As a physician and as well as a consultant in the pharma and healthcare space, I really appreciated this whole conversation and getting to learn so much about a number of different topics. It was fascinating and wonderful to hear about Dr. Yancopoulos' background, his many Columbia degrees, his journey at Regeneron, as well as some of the science and the issues around COVID and beyond COVID even, as well as some more even higher level and inspiring comments around how this really is a game of life or death for all of us, and we have to think about how we fight this together. It was particularly inspiring of a few points just to hear and learn about some of the complexities around the vaccines, the treatments, and even these Regeneron, these new Regeneron antibodies, and how we can even create some of these tools and some of these vaccines and antibodies, I should say, outside of the human body and the potential. So what a wonderful way to spend an hour this evening. On behalf of the college alumni community, I do want to express my deepest thanks to Dr. Yancopoulos, Dean Valentini, and our important audience, Columbia alumni, for joining us. We encourage you to stay involved and engage with the college community. Thank you, and have a wonderful evening.